as Megan said, I am a mucosal immunologist, and so I'm kind of coming into the interested in the world of breast milk from an infectious disease and mucosal surfaces type of focus here. So for a while now, I've been focused on late onset neonatal sepsis and bloodstream infections. And these, of course, are systemic bloodstream infections that occur particularly during the first few weeks of life. And they can be caused by a variety of bacteria, so both gram-positive and gram-negative. And they have a pretty nasty incidence of 10%, particularly within those infants that are born at a very low birth weight. And within that, they can have a relatively high mortality rate, and they represent a huge majority of all neonatal fatalities. So I got interested in this because while a majority of the causative pathogens that can be found in these neonatal sepsis infections are routinely introduced from the skin during like clinical practices, there's a big subset of these patients that will present, and the causative pathogen are things like E. coli or Enterobacter or Enterococcus. And these causative pathogens can be found in the gut of the infants days before an infection will set in. So it really suggests that there is a mechanism for these normal pathobionts that live in the gut and are colonizing the gut to translocate and break loose from the intestine and set up these systemic infections that can be pretty nasty to the infant. So my lab has really focused on this route of bacterial infection. And of course, since this is the Human Milk Club, one angle is really understanding how breast milk and maternal protection can help protect the infants against these gut pathogens and sepsis. So it's been well known and well observed that breastfeeding, at least partial breastfeeding, reduces the risk of sepsis. And we also know in the mucosal field that breastfeeding really reduces gut permeability in infants. And so when we start to take a look at specific components in breast milk and human milk that can contribute to this protection against gut permeability, we really started to move towards animal models, and I'll be sharing some animal modeling data because here we can start to formula feed animals, and then we can look at things like bacterial translocation to the mesenteric lymph node and the liver and see that there is increased bacterial translocation when you place animals on formula compared to dam-fed animals. And then you have a nice model to add back different components to really test the role of each individual contributor into milk to help protect against different pathogens. So I don't know if this is the paper that Megan was suggesting, but really one of the papers that kind of led to the found of my lab is looking at one particular component in milk, which is epidermal growth factor, which can be found in milk in increasing concentrations, particularly in colostrum, right around after lactation is initiated. So one of the papers that led to the founding of my lab really looked at this specific role of epidermal growth factor. And so we found that it can actively bind epidermal growth factor receptor. Its receptor is expressed on the infant's small intestine epithelium. So this is a model showing that now we're in the infant's, the offspring's intestinal tract. And the epithelial cells express the receptor for this ligand that's really only found in human milk at this time, or dam's milk. And this specific interaction here, if you trigger it on a type of cell called a goblet cell, this can improve gut permeability. So it really limits the ability of microbes to translocate and get across the epithelium. So similarly, if you have a lack of EGF because the infants are on formula, or let's see here. Oh, I can get to that too later. I'll answer that question later about the cell wall. Right, if you have a lack of EGF, and then we showed by knocking out the different components, a lack of the receptor, then you have this way for these bacteria to get across the epithelium and induce this model of sepsis. So we've been using this model of sepsis for quite some time, But we got very interested in what are some other components that can be contributing to protection within the lumen of the infant's gut. And there's a lot of good things, HMOs, of course, and things that feed the microbiota. But as an immunologist, I'm interested in things that have an immunological consequence in the infant. And so maternal immunoglobulins have been the focus of the lab now for some time, because as you guys all know, human milk is rich in maternal IgA and IgG, and these are two different isotypes of antibodies that can bind toxins and viruses and bacteria. They can be polyreactive for a lot of different shared components of microbes, and so they are really important for luminal protection from enteric pathogens. And it's kind of funny that we know this in the livestock industry for years and years and years, how important antibodies are for offspring. And we're only starting to apply this to human biology. And so this story really starts and focuses on how do maternal immunoglobulins protect against the neonate against enteric-derived LOS. So about 2 years ago now, a now friend and colleague, Chrissy Megley from the University of Pittsburgh, started this cohort in Pittsburgh and called me up afterwards. But she has this she's an OBGYN, and she had this clinical observation that maternal antibiotic exposure within 72 hours before delivery was associated with greater infant sepsis risk. And so she went to a historical chart review and looked at women that had premature PPROM, so premature rupture of the membranes. And then she broke this cohort into those that were received traditional antibiotics and those that received non-traditional antibiotics. Now, traditionally, most people that present with premature PPROM are going to be given ampicillin or amoxicillin or azithromycin. In the case of perceived penicillin allergies, it's then left up to clinicians. So this is where the non-traditional antibiotics come in. So this can be anything from a non-beta-lactam to a cephalosporin. And so what she found is if you compared those given traditional antibiotics to those given a non-traditional antibiotic, that there was a significant increased risk of LOS specifically when you compared it to other infant outcomes. And then let's see what else. If you pulled it out to cephalosporin, she saw the same increased risk of LOS. And so at this point, she called me up and she was curious if we could try to apply my model to figure out what was going on with antibiotic use and why would maternal cephalosporin or broad spectrum antibiotics increase the risk for sepsis. So meanwhile, my lab had actually fortuitously been doing some work on this, where we took mice that were pregnant and exposed them to antibiotics specifically during the week right before they delivered. And they have a gestational time of 21 days. So this is embryonic day 14 here in my little nomenclature. And so they're on antibiotics for 1 week. And then we looked at the amount of IgA and other antibodies 2 days after birth. And so when we put the mice on specific antibiotics, we saw this significant decrease in IgA when we gave them cephalosporins like cephalexin or ceftazidime. And this was only seen in their mammary glands. It wasn't seen in the serum of the dame. So it was something that was specifically targeting the mammary glands. When we looked next at the stomach contents of the pups, we saw the same decrease. So again, now we're seeing a decrease in the IgA measured in the pup's small intestinal and colon tracts here, but not in their plasma. So it's still able to get across circulation, and they still have no problem making IgA, but we do see this decrease within their intestinal tract, linking it back to their mother's milk. So then we specifically looked out at cephalexin, and we took these pups out to 1 week postnatal. So now they're 7 days old. And we again saw much of the same data. And I'm just going to kind of breeze through this because this isn't the interesting bit. But we saw a significant decrease in IgA in the stomach and the small intestine in the stool without affecting the serum. And this was, again, similarly seen in the mammary glands of the dams. But we didn't affect IgG in either the pups or the dam. So it was specific to IgA. Now we are placing these mice on antibiotics. So we looked at the microbiota by 16S sequencing. And what we found was the dams that were given the antibiotics really didn't have much of a microbial disruption within their intestines. So their intestinal microbiotas were very much the same, and they're in pink here. So by principal component analysis, we didn't see too much difference. And even the pups slightly overlapped, but what we did notice in the pups that were treated with cephalosporins here in these closed dots in blue, they really had this huge expansion of gamma proteobacteria here in this stacked bar plot in purple. And we noted that this was all due to commensal E. coli now within the intestinal tracts of these pups. And so then when we took this to our model of sepsis, we then challenged these pups with a gavage of a bloodstream isolate E. coli. So this is an E. coli that we got from a septic patient. So we gavaged the pups with it on postnatal day 9, and only the pups that were from moms that were treated with cephalexin, they had an increased amount of E. coli colonizing their intestinal tracts, and they had an increased risk of death in response to this. So that's the mouse data, but I just wanted to take it back because really excitingly, when we took this back to Chrissy, she had the ability to pull milk samples then from women that had premature PPROM, and then were given either no antibiotics or less than 1 day of antibiotics. And then we could also trace it back to, again, the mothers that were given ampicillin or azithromycin for the traditional regimen versus the broad spectrum regimen. And again, similar to the mouse model, in this human milk, now we see this decrease of IgA and IgM, but not IgG here. And so for this is really exciting for us because we saw this increased risk of LOS following antenatal cephalosporins and a decreased mammary IgA and IgM following broad spectrum antibiotics in the human data. And so we are thinking that this lack of IgA and IgM then will allow for increased pathobiont colonization, like what we saw in the pups. And then this will allow them to get closer to the epithelium and potentially have a way to translocate, like what we saw in the pups. So thinking as an immunologist, my next big question is going to be, how is this maternal antibiotics use perturbing the maternal intestinal microbiota? And how is the maternal intestinal microbiota driving the antibody and plasma cell trafficking to the mammary glands? But I think practically this has a lot of implications for antibiotic use and how a mom might do everything right and be breastfeeding, but still might not be providing sufficient IgA given other environmental factors. So this is just a little schematic showing how now we're thinking about maternal antibodies and then our original component of interest, EGF, in kind of tandem, where we have one component from human milk, the antibodies that are restricting these bacteria from the lumen or from the epithelium and keeping them in the luminal space. And then you have EGF, which has this really interesting relationship, receptor ligand pairing with the intestinal epithelial cells that prevent these cells from becoming a pathway from the bacteria. So there's some really nice synergy at play here. So that's a very brief chat about what's going on in the lab. And so this is a picture of my lab here with the people in blue highlighted who are performing the work, and italicized people have sadly left the lab and gone on to do other things. And I have some lovely collaborators here at Mayo Clinic in the neonatology world, Dr. Brombaugh and Levy. And then of course, this work is done in tandem with Chrissy Megley.